I've kind of put enumeration and biovolume in with identification, so I'm trying to compress this, and I haven't learned what is the optimum amount of information to throw at you yet. So if you'll bear with me on this one, we might find this lecture a little bit, I won't use the word tedious, we'll just use lots of information. Interesting, more interesting than the other side. So I wanted to talk then about the harmful algal bloom groups collectively, including the marine, and then what are the major approaches to identifying the taxonomy of cyanobacteria, and enumeration and biovolume. The enumeration and biovolume, I'm never quite sure how much, because many agencies want you to do that, many agencies don't want you to do that. So putting this in for those that find that we need cell counts and we also want biovolumes. So the terminology, which we're pretty much already set, but water blooms are proliferations of all those different shapes of algae, whether they're single cells, filaments, colonies, even macroalgae, seaweeds would be called a water bloom. Proliferation occurs when nutrients, temperature, pH, light are conducive to good growth, but only a few of the many phytoplankton species produce the toxins. So we have maybe 1,500, the new taxonomy would say maybe closer to 2,000 species of cyanobacteria. As far as I know, probably less than 100 are producing toxins. Now we add to that all the time. I've got 40 here, I would probably go more with 50, but we just haven't tested them. So it's not very many of those particular genera that produce toxins. Same with the marine. It's not the majority, it's very much the minority that produce toxins. But the proliferation of the non-toxic ones can also be considered a hab. So if you've got just a big pile of algae and you're causing stress and killing fish and closing beaches, then why not? That's also considered a hab. All states in the US and countries worldwide can be affected, and those blooms tend to be based on their color. So we know about red tides, brown tides, and now of course we have our blue tides or blue-green tides or whatever you want to call them. Those 8 algal divisions that I just mentioned, of course the cyanophytes, the new term for cyanobacteria is not Cyanophyta, the division name or a phylum name, depending on how you approach it. It used to be Cyanophyta. The new taxonomy has the term Cyanoprokaryonta. And of course that's to take the phyta, the plant recognition, away and replace it a little more with the microbiological prokaryotic approach, Prokaryonta. So these used to be the blue-green algae or cyanobacteria, 1,500 to 2,000 species. Contrast that to everything else that's an alga, they're all eukaryotes. So the one that's we consider to be the phylogenetic route to higher plants would be the green algae, about 6,000 species. None of those are poisonous. Likewise, the charophytes, the stoneworts or brittleworts, sometimes they're placed with the chlorophytes. So that's chara, the stonewort that you have in some of your alkaline lakes around here. The chrysophytes, the golden algae, this includes the diatoms, and of course we know that we have a toxic species of marine diatom, Pseudonytzia, which we'll show you in a bit. But you 6,000 species, but that's the only one that's toxic in that group. There are quite a number in that group, the golden algae, that produce taste and odor issues for drinking water supplies, but not toxins. Pyrophyta dinoflagellates, that's where we've got most of the marine toxin producing algae. And the euglenias, which are a small group and found a lot in really nutrient-rich water. So your wastewater ponds and your stock ponds that receive a lot of nutrients often have euglenia. But there are none of those that are toxic. And then the last two are considered the marine macrophytes, your big kelps and red algae and brown algae. So, yeah.
The pseudonychias in Chrysophyta?
Chrysophyta, yeah, the diatom. Pseudonychia, the ASP, amnesic shellfish poison. Well, being an integral part of food chain, you can imagine that this is part of the issue with calves as they vector as plankton into smaller invertebrates and then bigger invertebrates and then fish into mammals and fish and mammals into humans. So that's the issue. They're all considered primary producers because they fix atmospheric CO2 and produce products. Already have this one, so just again, what I'm going to do now is quickly go through and show you the primary organism producing these toxins, and including ASP, the diatom, and then move on to the cyanobacteria. So I'm going to start with who produces PSP and then DSP and NSP, so on. Just one or two slides. So the dinoflagellates. We have freshwater dinoflagellates. In fact, this time of year, it's quite often to see dinoflagellate blooms preceding the cyanobacteria blooms. I've seen this now in Oregon. We've got some good dinoflagellate blooms, and they're being displaced by cyanobacteria blooms as the summer progresses. So there are diverse unicellular filamentous protists. Dinoflagellates are strange because they can be all the way from parasitic to photosynthetic. There are parasitic dinoflagellates. And there are those that flop back and forth between being heterotrophic, needing preformed organic material to live, and being fully photosynthetic. So they cover a broad spectrum. Some of them are bioluminescent. As I said, some can be parasitic. And of course, some of those that form the blooms become golden brown, brown, or red tides. Eating the contaminated fish or shellfish results in syndromes like the PSP, DSP, ciguatera poisoning. So first, the PSP. It started in California, the very first cases, but it's become more established in the New England states and Alaska as a continuing issue. A number of dinoflagellates there that produce them. The symptoms are gastrointestinal and neurological, and they can be life-threatening. Again, being neurotoxins, if you ingest the shellfish containing that toxin, the first thing you feel is, oh, this is fun, I'm tingling. But that doesn't stop there. Then you get numbness and drowsiness and fever, and then respiratory arrest. Correct? Yes?
So I'm curious, you said it started in California and then it's kind of moved to the...
Well, it doesn't, yeah, it's being displaced more by this diatom blooms here, but the original source of the red tide for the saxitoxin was from California.
And is that something that is, a recent phenomenon, or is it something that's always occurred that was kind of single?
Well, I think the red tides have a phenomenon been here for a long, long time because native populations recognized them hundreds and hundreds of years ago. And they established protocols for the times of year in which you avoided collecting shellfish to minimize exposure to the toxins. And that's all the way up California, Mexico, all the way down South America, all the way up to Alaska. So those are well established. So they've got to have been here a long, long time. But, they're changing with changing climates and things.
What is the freshwater brackish marine requirements for these again?
For the
ones that can produce the paralytic
paralytic? Well, they're truly marine. So there are salinity regimes, but they're all right around whatever, 32 parts per thousand, roughly in that range. And there are influences of salinity on growth, but it's not the major issue. They are truly marine. The main one is this guy. Alexandrium. This is one of the primary producers of saxitoxin. And these are called the armored dinoflagellates. Each one of these little structures here is a thecal plate, like an armored plate, like a covering over that you put on if you were a knight in shining armor, made of a protein, a hard protein, and a pectin material. This groove right here, this is an electron microscope, but normally they'd have flagella, two flagella, one wrapped around here and one here for motility. And this is a single cell, but they could be 50 to 100. They're quite a bit bigger than this, cyanobacteria cell. But that is one of your primary PSP producers. The DSP producer, diuretic shellfish poison, as the name implies, it's more of a diuretic problem. DSP distributed worldwide. It's more of an issue in Europe and North and South America. And the toxin that's produced is called okadaic acid. The interesting thing about that toxin is it has the same mechanism of action as microcystin. It's a protein phosphatase inhibitor. But okadaic acid acts in the gut and leads to diarrhea. Whereas microcystin has a transport mechanism that takes it into the circulation and into the liver where it causes its effects. So that's a fascinating sort of thing, but they both inhibit protein phosphatases. Different groupings, but the same. This is what Dinophysius looks like. Again, that's a single cell, and there would be a flagella around the top part of that cell. The neurotoxic shellfish poisoning, again, we're back to kind of a neurotoxin. It was first recorded in Florida, Gulf of Mexico, but it's now found pretty much worldwide as well. And it's associated with ingestion of contaminated shellfish, but the symptoms are milder. They don't usually lead to a lethal situation. But they're more responsible for fish deaths. Hundreds of tons of fish can die from these toxins. And the toxins have been identified and they're called brevetoxins. They can also be present in aerosols. So the red tide in the surf producing an aerosol, people exposed on the beach. So they actually close and post beaches in Florida based on the red tide. And they have respiratory centers where you can go in and get air-conditioned air when you're at the beach, if it's a situation. This is also the toxin that's been responsible for deaths of manatees and other large mammals in those areas. The genus is called Carmichael, and they are brevetoxins. They're gastrointestinal and neurological and respiratory if they're inhaled. This is just an example of a Carmichael bloom. This again looks like a red tide, and these are dead fish floating around. And one of the organisms looks like this. So this is, again, a single cell of Carmichael, and in this case it's a living cell, so you can see the flagella. But the flagella wraps around the sulcus and trails and leads to motility. But again, one cell. Ciguatera, fish poisoning. Now the previous one, like PSP, we said shellfish poisoning because the vector is usually shellfish. This toxin is largely a fish vector toxin, so it's called fish poisoning or ciguatera. It is the most common of those marine toxins from algae. This 50,000 people is probably no longer valid. It's probably much more than that, but it's especially common in the Pacific and the Caribbean. So they're all different kinds. The main one is a genus called Gambrodiscus. They produce the toxins of ciguatoxin and mitotoxin, ciguatoxin being the main one. They are neurologic and gastrointestinal. So even though they're neurologic, they're more gastrointestinal of a toxin. So they cause severe diarrhea, abdominal pain, nausea, dizziness. The other interesting thing is this reversal of hot and cold. So cold water feels hot, and hot water feels cold. Ciguatera toxin is long-lasting. If you have a bad dose of this, you don't die, but you have these symptoms sometimes months to years. So it's long-lasting. There are some treatments, though. I'm not sure how these work, but vitamins and antihistamines and steroids can alleviate the symptoms long-term. They can vector through almost any reef-feeding fish. So they grow on the reef. They aren't a bloom former. They grow attached to the reef. Fish feeding on the reef concentrate the toxin, and then you ingest the fish. And I had a graduate student once who went there and got Ciguatera. So sometimes you have to monitor fish sold in markets because they have dipstick assays and immunoassays for testing the flesh of fish sold in markets where this is a problem. And there are strict regulations about importing fish that have come from these Ciguatera-infested areas. And Gambardiscus looks like this. Again, a single cell, and that groove is where the flagella resides and provides the motility. And you can see little lines. Those would be the lines for the fecal plates. They aren't as prominent as in the Alexandrium, but they do have fecal plates as well. So finally, those are the dinoflagellates. The one diatom producing, which is a problem here in California and in Oregon and Washington, the amnesic shellfish poisoning. It's a marine toxin disease again. We don't seem to have found it in any of the diatoms that have been tested in the freshwater environment. And the genus of diatom is renamed. It used to be called Nitsia, but better identification with electron microscopy has sort of changed its terminology. It's now referred to as pseudo-Nitsia. I guess that means false Nitsia. And there are a couple of different species. Pseudo-Nitsia, Pseudodelicatissima, and pungens are the two main ones. And the micrograph on the right shows you an electron microscope view of the, diatoms are beautiful organisms. They have all this ornamentation because of the silica frustules that they produce. And the ornamentations, those holes and the arrangements of the holes, are what you use to do the species and variety identification. So you really have to do electron microscopy to do the identification correctly. The slide on the left is just a light microscope of the film of the cell itself. That long, slender cell can be a couple hundred microns long. There's a scale there, at least about 100 microns long, and that's one cell. And then if you magnified that cell, you'd see these ornamentations, the so-called poroids, which is what's needed again to get the right species identification. And again, it's just a marine, but it's just the one that's in the Gulf of Monterey Bay, sea lions, because this one will vector. So things like sardines accumulate the toxin, which are then consumed by marine mammals. So it's more of it's not a human poisoning, I don't think, although it could be if you ate shellfish, but we restrict and test for shellfish. So. But no one's really died from this toxin. But again, it's a debilitating toxin if you're exposed to it. Some of the marine programs then, biotoxin programs, NOAA, of course, National Oceanographic Atmospheric Administration, which has received most of the funding for marine toxin work over the years, has a website and information. Woods Hole, which is where Don Anderson has set up his lab, he's the fellow that coined this term, harmful algal bloom, remember? They have a very good website, and he actually has the authority to have the national harmful algal bloom website that Congress has mandated. So it's probably the more useful website for HABs. Again, mostly marine, but this one also includes freshwater. The golden algae, Primnesium, again, as I mentioned, Primnesium is now showing up in your lakes here, some of your reservoirs. It's a brackish water flagellate. It's completely different division, producing these hemolytic cytotoxins called primnesins, leading to mass die-offs of fish and sometimes clams and mussels. First identified in the 1970s, more of an issue historically in the US in Texas, so they have the biggest program and they have probably the most useful website for Primnesium. An example of this is Lake Granbury in Texas. This is a massive fish die-off, so thousands of fish in this. In this case, it's a river and a lake. It's a reservoir, and the salinity increased enough due to contamination that led to these Primnesium blooms. Okay. The toxin structures were not known for a long time. They've only been known for about 10 years now, but they are published. So, cyanobacteria. Well, as I implied earlier, they've been around a long time, but in terms of events that sort of drive public policy and concern, Lake Erie was probably the first in the 70s. Lake Erie was not dead. Lake Erie was very much alive, just a little rich in nutrients. Managed to reduce those blooms with point source nutrient restrictions. Non-point source non-restrictions have led probably the most to the resurgence of the blooms beginning in the mid-90s. This is also a reservoir that I looked at that drove the situation in South Africa. This is a reservoir. I would put this in because, again, to emphasize what happens when the cells start to die. You get the blue-green hints. And that blue-green color is an accessory photosynthetic pigment called phycocyanin. So, as the name implies, a blue pigment produced by an alga, phycocyanin. And most cyanobacteria have them. They also have something called phycoerythrin, which is a red accessory photosynthetic pigment, giving the color to the cells, and also responsible for part of the photosynthetic process. Phycocyanin, however, is only produced by cyanobacteria. So phycocyanin probes, if you go out and buy one of these YSI sondes, or there's one in here.
In the cardboard box.
Oh, this cardboard box is phycocyanin fluoresces, and so there's a little fluorescence detector. Maybe Eric will talk about that. These are some kits that kind of went with sampling. I'm going to maybe show them in the lab this afternoon. Eric brought them. But this is a Turner probe. Turner has been around a long time in terms of fluorescent detectors. And some of these accessory pigments, phycoerythrin and phycocyanin, will fluoresce at certain wavelengths. So if you can detect phycocyanin, then that's a surrogate for cyanobacteria. Because if you just go after chlorophyll, maybe you've got chlorophyll in other algae. So if you want to just detect cyanobacteria, phycocyanin, and that's the blue-green pigment. So that's a good point. Bring that up again. Yes?
Are you going to get into any other sensors used with like the Turner, the EXO, the YSI, anything like that?
No, I don't. I mean, they are. Hydrolab and YSI produce the more popular common sondes, and both of them have incorporated the Turner probe into their sondes, so you'd have to buy it as a separate probe. So when you buy a YSI probe apparatus, it'll have a temperature and a diode and a turbidity and chlorophyll, and maybe you add the phycocyanin probe. But those are the standard ways of monitoring for them. Does that kind of answer it? We don't have time to
Yeah, I was
show the probes.
our group, I'm just but I just, if you don't mind me
Yeah.
taking a moment, we have a monitoring program throughout the Delta. We have nutrient sensors and green algae,
All right.
YSI exos, so if any, I'd be interested in talking to anyone. I don't know the best venue to introduce that.
Sure.
But,
This afternoon.
certainly high frequency measurements are, they don't duplicate discrete sampling, but they're very valuable. Because you can look at everything on real time, everything's online, the USGS website, so
Right.
that can be of use if there is a bloom. It sounds like a lot of people are working outside the Delta, and monitoring programs take quite a bit of money to start up, but they are worth their weight when they are functional.
Oh, yeah, yeah, they are. They do require maintenance. But, yeah, so, yeah, the standard way of, setting up monitoring other than just sampling is to have some sort of SON system, and the main ones are Hydrolab or YSI. YSI markets a little bit more strongly, and because of their past history with their probes, they're probably a little more used. And they have very good webinars on site that, on their website that show you the probes, and their marketing is a little bit better. As an aside, as long as we're talking about that, I lived in Yellow Springs for 25 years, so our lab actually developed the phycocyanin probe with YSI. We did all of it. We did all of the testing of their prototype probes with our algae samples. So in fact, the YSI phycocyanin probe was developed with toxic microcystis as an example. So, anyway, but thanks for bringing that up. So microcystis, now kind of moving into the taxonomy of the cyanobacteria. Again, put this out, it's the sort of the main one people think about. Each individual cell in this colony, remember these are single cells that clump into a colony, and they're all held together by a matrix, a polysaccharide matrix. And the shape of that matrix is the shape of the colony, and the shape of the colony determines the species. They're all around 4 or 5 microns in size, the individual cell, so that a colony can be a few hundred microns in size. And then colonies can clump together as well, so you can actually have clumps in your lake or reservoir that can be the size of golf balls. But if you pick them up, they won't hold together. But you can pick up small clumps and they will hold together. And those clumps are many, maybe a few hundred colonies. But an individual colony is a couple hundred microns, individual cell, 4 or 5 microns, microcystis. Well, on the other sort of extreme in terms of their morphology, this is a Lingbia, and it's a mat-forming benthic freshwater species that produces saxitoxins. And this is actually my son's hands. We were working on a project on the Tennessee River, which had all of the macrophyte populations had been displaced by this bloom of mat-forming Lingbia, and the Tennessee Valley Authority wanted it investigated. They wanted it gone because bass fishermen were not happy anymore. And we didn't know if it produced a toxin, but we tested it, and sure enough, we found that this was producing saxitoxin. So that was the first evidence for freshwater Lingbia producing saxitoxin. But this is my son holding mats. The mats all grow on the bottom, but the mats, when they produce oxygen, oxygen builds up in these mats, and the mats literally lift up and start to float in huge rafts. And these rafts can literally be tons of material, so it's easy to collect. They tried all kinds of things. They brought in sterile carp, and sterile carp didn't like eating them very much either, and then they had sterile carp to worry about. But often with situations, you wait long enough and it goes away, or you get used to living with it, and I think they've gotten used to living with it. Another extreme habitat, just to point out where cyanobacteria grow, this is a little colonial 4 to 8 celled colony organism called Gleocapsid, and all this dark red material, if the lights were down it'd be darker, but this is dark reddish black. This is all Cyanobacteria Gleocapsid growing at high altitude. So they can they have that, this reddish color would be phycoerythrin, and that absorbs UV as well and protects the cell, because at high altitudes you'd have a UV toxicity if you weren't careful. So the white patch is a snowpack. As another cute example, where can you find blue-greens growing, just to emphasize. Here they are growing on the hair of a polar bear at the San Antonio Zoo. Polar bears have guard hairs, these two guard hairs that are highly insulated, and the algae actually grows down inside that. Of course, in a zoo in San Antonio, you might expect that you wouldn't find them growing algae in the Arctic, but at least in this situation they are. And they don't harm the polar bear in this case. Often we have cyanobacteria as symbiotic. This greenish, dark green color are colonies of unicellular marine cyanobacteria growing on the surface of a sea squirt or marine tunicate. So this is a symbiont. We were studying this because this tunicate produces a solid anti-tumor inhibiting anti-cancer compound called didemnin. And the group of organisms are called trididemnins. So this compound is called didemnin. And I was asking a question, plus to get the diagonal was nice too, but were the cyanobacteria producing this anti-cancer compound? And what we discovered through some labeling experiments was that indeed the cyanobacteria produce the precursor to the anti-cancer compound, which is then completely produced by the tunicate. So in reality, the tunicate produces it, but not without the presence of the symbiotic cyanobacteria. So that's an interesting study. And that compound is actually made through phase 2 trials and is commercially used as a solid tumor inhibitor, cancer. Okay, taxonomy. Two approaches, the botanical code and the bacteriological code. As you might appreciate, before we learned they were prokaryotic, we approached all cyanobacteria taxonomy using the botanical approach. Requires the specimens be described in Latin, and the specimen dried down and pressed between papers and stuck in a herbarium, and that's this type specimen. Well, the bacteriological approach, ruled by the bacteriological code, requires a culture rather than herbarium specimen. And it has different approaches, more of a physiological biochemical approach to taxonomy. The botanical approach obviously has limitations. Dried specimens 100 years old in a herbarium probably are difficult to identify. But on the other hand, an interesting side point, test those and they all produce toxins. So the toxins are stable at least a couple hundred years. I worked at the Pasteur Institute and the National Herbarium in Paris for a few months, and we tested specimens out of that material, and indeed they produce active microsystems. We don't know what there was originally 100 years or 150 years ago. These were all specimens collected by the early French taxonomists. So the bacteriological approach is coming along, and in fact was proposed as being the only way. Problem is, if you collect a sample out of the lake, put it under the microscope, what are you going to do? You're going to have to grow it, test its temperature and its lipid content, and plate it like you would a bacteria and look for color reaction. That doesn't work. So you still have to fall back on the old classical botanical morphology. And that's what it's come down to. The new taxonomy really incorporates the botanical approach with the genetic approach. And I'll get to that in a little bit. So the main characteristics that you have to work with cyanobacteria is that they have chlorophyll A and they produce oxygen. They don't have chlorophyll B like the chlorophyta division does. So they aren't, true plants. But they have chlorophyll A, so you could measure chlorophyll, which you can do with the sonde that you buy from YSI, and that's perfectly fine. The accessory photosynthetic pigments, so the phycocyanin and phycoerythrin, become more specific to cyanobacteria. Hence, having a phycocyanin probe stuck on your YSI sonde really enhances your ability to segregate and focus on cyanobacteria. Their food storage is glycogen like bacteria, so that's not a really good taxonomy clue. But it also does mean that doing a starch test doesn't work because they won't have starch. And of course, one of the things that's main about them is they have no sexual reproduction, just like bacteria, they have binary fission. And as a consequence of being prokaryotic, they have a 16S RNA, which is extremely important, as we're going to come to learn, as opposed to 18S RNA that your eukaryotic organisms do. So the real things that we're focusing in on with cyanobacteria for taxonomy and identification and classification are these accessory photosynthetic pigments and the 16S RNA in terms of the new stuff. So we can really then approach this taxonomy morphologically, physiological, and biochemical and genetic, and I've highlighted the red, which morphology and genetic, which is really where it's headed now with cyanobacteria. Some of the morphological terms that you're going to find in this taxonomy key that you're going to have to learn to identify cyanobacteria. We talk, and the first term, thallus, is really something for plants in general. If you're holding a rose in your hand, that's a thallus. That's the plant, whatever it is. So if you're holding a filament in your hand, that's a thallus. So thallus just refers to the basic shape of the organism, whether it's unicell, filamentous, or colonial. The sheath around that is peculiar, even though it's not peculiar to cyanobacteria, it's peculiar to prokaryotes. That slime layer helps hold the cell together and protects the cell. Then branching is important in the taxonomy of the cyanobacteria. If we have filaments and those filaments branch, that's going to be a clue that this is a different, actually probably a different family altogether. Most of the ones that produce toxins don't branch. There are a few, but not many. And those filaments branched or unbranched, and the branching can be false branching or true branching. And I want you to hold that thought. If I'm a tree and I produce a branch like that, it's attached to the main stem. That's true branching. But inside that sheath, that slime layer, you can have a filament, and the sheath actually keeps the filament breaks and starts growing sideways. It's not attached to the main. So that's false branching. And that's a key taxonomy to different groups of cyanobacteria. Not so important for toxic ones, but is one of the key characteristics. And then, of course, the specialized cells, the heterocyst and the akinete. They are important because they clearly throw most of the ones that produce heterocysts and akinetes, the nostochales, they contain members that produce all of the different cyanotoxins. Microcystis only produces microcystin. Anabina, or now called Dolichospermum, can produce all of the different cyanotoxins, depending on the species and so on. So that seeing those heterocysts, and may or may not, you don't have to see akinetes to identify it because they always have heterocysts, but they're only going to produce akinetes later on in the season as they're going into dormancy, or early on. They don't necessarily produce them during the active growing season when you have blooms. So it's really the heterocyst that's a giveaway for identifying many of the toxic ones. Now, if we were looking at biochemical sort of identification, we might look at these things like carbohydrates and lipids and proteins and so on. That's really not important anymore. It would be if we did the bacteriological approach, but we don't worry about that. But we are worried about these RNAs, especially the 16S RNA. We don't worry about DNAs when it comes to cyanobacteria taxonomy. It's really this 16S RNA. And then, of course, the pigments. So that 16S RNA is universal and highly conserved, making it useful for taxonomy. And it's also useful for identifying cyanobacteria. So 16S RNA. Well, how do we work with 16S RNA? Well, if you were my age, you never did work with 16S RNA because all of the genetic approaches are probably within the last 20 years. So you never had a molecular genetics class as an undergraduate when I was an undergraduate. But 16S RNA is extremely important anymore, and the techniques of reverse transcriptase and cloning are older methods replaced almost exclusively by PCR, or polymerase chain reaction. So, polymerase chain reaction allows you to make these taxonomic trees. This is an example, and a phanozomenon is placed there in the middle, and depending upon the distance on that tree, you are less and less like that species. And so this is how it's done. And of course there are hundreds of phylogenetic trees depending upon the group you're working with. All based on others, there are a few others, but 16S RNA is the main one. Now some of the other PCR genes that are used occasionally, the nitrogen fixing gene, remember heterocysts fix nitrogen and they have a certain gene sequence. So sometimes those NIF genes or nitrogen fixation genes are used taxonomically. There's another one for the phycobilizome. Phycobilizomes are the structures that contain those phycocyanin and phycoerythrin pigments. So the genes in those subunits are used in taxonomy. And of course, more recently, the toxin genes themselves. I just listed here MCY, which is the toxin gene for microcystin. There's MCY, ABCD kind of thing, roughly equivalent to the number of amino acid units in the molecule. And certain of those genes, I think the one that's used most now is the microcystin B gene to determine presence of microcystins. At the workshop in San Marcos, there was a company who's now adapted the new test kit for the MCY genes, and it has a probe set up. You actually test for all of them, all the ones we have genes for, which is saxitoxin, cylindrospermopsin, microcystin, I guess that's it. And the anatoxin gene is coming along. So you mix those all together, make yourself a probe kit, and you can identify that. And so that's where that's headed, the genes. So some of the references for taxonomy. AWWA identification in water supplies. As I said, the new taxonomy has just been published the last few years, and this was mostly done by this fellow in Czechoslovakia, Yuri Kumarek, and his colleague Anagnostides, who actually he just passed away recently. But the very first volume of that new series, the Crow-Cockales, all three of these volumes, 1998, 2005, and 2013, are big. They're about that thick. The last one is well over 1,000 pages. So unless you're really serious about taxonomy, I would stay away from those. But that's what there is if you want the definitive taxonomy anymore. About 2,500 pages of small print. But it's a Herculean task. They're to be congratulated. But as I said, it incorporates the morphological and the genetic primarily. Now, much smaller versions of that have been condensed, and one that I want to show you this afternoon is Andrew Chapman's chapter in the AWWA manual, where he has a little 3-page key, and I'll have keys for you to practice with this afternoon. There's another nice book put out by, this is the group in Europe, it's a picture book, this one, Manual of Aquatic Cyanobacteria, beautiful photographs. And this was put out by EU, but you have to pay for that. But it's a nice picture guide. But I've done the better thing, and that is to put up the picture guide on the web. That's the other PDF download for you, which is a new one, a USGS sort of picture guide. So this one, Rosen and St. Amand, Anne, Field and Laboratory Guide to Freshwater Cyanobacteria. This is what I'd like you to download and kind of have at your perusal when you're working with cyanobacteria. Very nice pictures. It was designed for cyanohabs, but it was done by the USGS. And it's all downloadable, so that's good. And then if you really want to get in more to the taxonomy with beautiful pictures as well, but not get those Kumarek books, which have no pictures except drawings, then this new one, Robin Matthews, she is at University of Washington, which one it is. It's not Washington State, but anyway, one near Spokane. Her first volume, called Book 6, Freshwater Algae, Northwest Washington, Volume 1, Cyanobacteria. It's a beautifully illustrated, and it's all free, downloadable on that website. And that's one I recommend, is having a more advanced little taxonomy with pictures, and then having the USGS one as well. It's about all you need really now, and they're free, so even better. So Chapman's cyanobacteria organism relies on something called a dichotomous key. And I just put up the first page of this, but to remind you, if you ever took a plant taxonomy course, it's always dichotomous, isn't it? Yes, no. You're given a characteristic, you hold up the leaf and you say yes or no. You say no, then you go to the next one on the list. Same thing. So this one, though, kind of identifies the taste and odor and toxin producers in the key, so that's helpful. So the first thing you would ask when you look at a specimen under the scope is, are those cells solitary, grouped in clusters, not forming mucilaginous colonies, ovate or cylindrical? If the answer was yes, you would have Synechococcus. If you would say no, then you move to the next one, cells organized in colonies or in trichomes, cells not solitary. If it's yes, then you go to 2A, cells in a few to many colonies, and a few would mean handful, half a dozen, dozen, and many would be hundreds to thousands. And then if the answer is yes, you go to 3, and presumably that's going to give you that particular genus, Cyanodiptyum. And cells not embedded in mucilaginous, the mucilaginous strand refers to that slime layer that I've talked about. You'd go to 4 and you'd work on it. So I'm going to give you a specimen this afternoon and encourage you to take a key and sort of walk it through and see if you can come up with that organism. It gives you the genus and tells you. In fact, I'll tell you, the one that I want you to look at is going to be an Anabina, see if you can walk through the key. This is just part of the first page. There are only 3 pages to the kit, or to the key. So, what about enumeration and biovolumes in the remaining time? And this is where I'm still having trouble how much to give you. But if you want to count cells, how do we count cells? I guess is what it comes down to. Well, you can't read this that well, but this is an RFP that was thrown at me by the city of Salem, which is our capital in Oregon. And they wanted a response for a proposal to do a monitoring cell count of cyanobacteria in water supplies. And they wanted all these things, analyzing freshwater algae, including planktonic and attached, that's the paraphytic, ability to analyze samples for heterocysts and aconites, conduct standard analysis and preserve samples, and they're using Lugol's as a preservative. They wanted identification to group and species, density, percent, biovolume, and so on and so forth, and they probably didn't want to pay very much. So this is sometimes what you see. So this is everything. So this is why I say, maybe you don't want to do biovolumes, but maybe some agency does. So I've tried to kind of run through this. Some of the typical questions then, in order to obtain a statistically sound cell count, what's the lowest number of cells you can count? How many are you going to count? Is counting 100? Is counting 10? Do you have to count 1,000 to get a statistical significance? That's what I'm going to tell you fairly quickly. After the lowest number has been reached, is it correct to assume that this is when sedimentation? In other words, if you have something at a certain level, let's say you only have 100 and you have 90, and that's below 100, then maybe you have to concentrate the sample so you do get 100. So that's the first thing you're asking when it comes to enumeration. Do I have enough to get a statistical significance, or do I have to concentrate the sample? Some of the references that are available for you, again, the WHO book, which I have a copy of, actually, the Sampling and Identification Methods and Strategies, that AWWA manual, same one that the key is in, and the standard methods, they all have enumeration and biovolume discussions. But what I'm going to try and do is summarize a bit for you. So here's a Lyngbia, straight chain filament. How do we count it? Am I going to have to count all the things that are attached in that, or do I count a certain number?
You count the number of filaments, not the number of cells.
Yeah. I'm going to, gee, you spoiled it for me. No, you didn't. And then something like this. Here's microcystis colonies. Many colonies clumped together. How are we going to count those? What she's referring to is natural units, and that would be the colony. But those clumps are hundreds of colonies. You still have to separate them and determine how many of those to count, even if you're only counting natural unit. So a natural unit is either a single filament, a single colony, whatever the organism grows as opposed to counting 10,000 cells in a colony and then having to count several colonies. But here's an aphanizomenon. Each one of those little curved flakes is a colony, and they're macroscopic. And in fact, they look a bit like this under the microscope. Each one of those flakes is a colony. Each one of those flakes would have several hundred, maybe 1,000 or 2,000 filaments. Each filament has a few hundred cells. Why wouldn't we probably want to decide that let's just count the colony, not the cells? But I'll talk about which one. So, again, don't read all this, but basically this is following through the rationale of what to count for an aphanizomenon. That a single filament is a typical size, and there are 1,000 micrometers in a 1 millimeter, so the AFA arranges itself in flakes. There may be 230 filaments lying parallel to each other, and then you've got 3 dimensions too. It's not just length and width, it's depth. So when it all comes down to it, we could have as many as 500,000 filaments in that one colony, and each filament is going to have a couple hundred cells. So you don't want to count those. So that's where we approach the natural units. So when it comes to enumeration then, we can quantify by counting, or the counting units available to you for quantification are the natural units, or perhaps total cells, or sometimes aerial standard units. If you've got a boss who's insisting you count cells, then maybe you're going to pick a standard aerial unit and just grid off a piece of that colony and count the cells in that piece, and then measure roughly the shape of the colony, and then you can estimate the number of total cells in the colony. But many places just deal with natural units, and that works okay because most of your cyanohabs are colonial, aren't they? Microcystis, Anamina, Aphanizomenon. Of course, oscillatorias or Planktothrix are not. Lyngbia is not, but a number of them are.
Dr. Carmichael, how would you deal with Eotrichia?
A natural unit, I would count the colony. And I have an example of that here too. So, in the standard phytoplankton counting units in the AWWA manual, they discuss enumeration as total cell counts, where you would count an individual cell and you report cells per mil. Or you would do natural unit counts where one organism, which is any unicell or colony, and you'd have units per mil. Or you might do an aerial standard unit count, in which this case is the standard unit is 400 square micrometers. And then you would also report that as units per mil, but report it as aerial units. And these are all acceptable limnological methods that, you know, your agencies, and you can explain something and presumably some other agency will be able to say, oh yes, I know what you're talking about, and we can compare apples to apples and oranges to oranges that way. Okay, well, what about concentrating samples? In concentrating samples, when do you need to concentrate? Well, basically when you have less than 10 to 15 cells in an undiluted count, cells or units, then you would need to concentrate. So that's the rule of thumb. You don't want a centrifuge, you want a filter. And the chamber that you concentrate in traditionally is called this Udermal chamber. And an Udermal chamber looks like this. You can buy them. It's a platform, and the cylinders are various sizes. So you have 5 mil, 10 mil, 20 mil, 50 mil, 100 mil. So you select how much you're sampling. You pour it in there. And these can actually be vacuum filtered a bit with these tubes to speed it up. But normally what you do is set these up in the afternoon, go home, come back the next day, and you filter it by settling down onto a membrane. Then you peel that membrane off as these cylinders come off, and you place that on your microscope, and they're transparent, and you do your unit counts or your cell counts. So this is the standard way of sort of concentrating samples, and it's used across the spectrum. So this just explains an example with the Udermal chamber. They're not expensive. The next decision to make is what kind of counting chamber do you take that, you peel off the Udermal, place it into a counting chamber, and then put that on the microscope. And it kind of depends on what you're after, you know, density, consideration of particulates and color and size distribution and so on, and how expensive your microscope is and how much time you're allowed to do this. But the counting chambers that are traditionally used are the following. The Sedgwick rafter, which holds an entire mill, and the one on your left is a gridded one, so maybe you're only going to count certain grids and then calculate based on the total volume. Or the bottom one is called a nanoplankton counting chamber. Phycotech, which is Ann St. Amon's company in Wisconsin, she calibrates a slide. She has a fixed volume on there, and each one's slightly different, but she calibrates that volume. And when you buy that chamber, you're given the exact volumes, 0.00567 milliliters or something. So you count everything in there, calculate it back to a mil, and then you've got units per mil or cells per mil. So those are, you know, counting chambers. Another one is called a Palmer-Maloney, which used to be used an awful lot, and this was used with something called a Whipple ocular micrometer. I don't know how much that's used anymore. I use the bottom one, called an improved Neubauer. It's actually devised for blood samples and things like that in hospital environments. But I like it because of its standardization. It's called the Improved Neubauer Hemocytometer. Hemocytometer implying blood cell counts. But it works really great for algae. I'll come back to that in just a second, but this Whipple ocular grid looks like this. So if you're using a Whipple counter, you would count maybe everything down the middle and across the side, and then you would average that across the whole specimen, and then you'd have a volume. With the Neubauer, this is what the grid looks like. And each one of those grids here is 1 millimeter on a side, or 1 square millimeter. So I count everything in 1 square millimeter, and then I do my calculations. But I'll count 4. I'll maybe do 1 here, and 1 down here, and 1 up here along this, so that I get an average of everything. And then I'll use that and I'll calculate cells per mil or units per mil. And I'll count them roughly in this direction, you know, being consistent. You want to count them in a consistent direction. So those are your hemocytometers. So, you know, the question of how many to count. If you have a density greater than equal to 10, that's what you need per taxa for a 95% confidence level. So as long as you've got 10 organisms you're counting, then you're okay. If you're doing units, you need 400 for 10% precision and 100 for 20. So as long as you're dealing with about 10 per field, per taxa, you're okay. So there's that take-home message. A couple of quick ones on biomass. Well, okay, here's a summary slide based on a workshop that we did. So you settle or filter the samples in that Unimol chamber, counting 400 natural units for 10% precision, count cells in 10 natural units for cells per mil, and that will give you the proper statistical significance. So what about biomass? I don't know if anybody works with biomass, but sometimes you're required to. Why do you even want to use biomass is another question. But a couple of things about biomass. One of the nice things is that biomass assumes a specific gravity of 1. Cells have a specific gravity of 1, so as long as you multiply by 1, you're fine. And sometimes you're concerned with carbon estimates, but that's a whole nother question. So if you're just wanting biomass, in other words, biovolume, biomass. Biovolume allows you to estimate the importance of individual taxa. Obviously, if you have small cells and big cells, the biovolumes, and you're counting cells, you got 10 cells, but what if one of those taxa is 100 times bigger than the other cell? Obviously, it's roughly 100 times more important to the total population. So there's the main importance of biovolumes. So you're calculating the volume of the living protoplasm. And to do that, the key simple thing is, what's the basic geometric shape? Do you have a cell that is round? Do you have one shaped like a cube, or a cone, or a cylinder, or a sickle? All of those are basic geometric shapes. So what that tells you is I don't have to measure the size of the cell and determine the volume. I can take a geometric shape. And taking that geometric shape, whether it's a sphere, there's a standard formula for it, pi r squared or length times width and that sort of thing. And they have built tables for all of the taxonomic genera based on those geometric shapes. So all you have to do is go to a table and look up the biovolume for that particular species and genus. And if you have cell counts already, then you just multiply them together, and that's how you get biovolumes. So it's really not as bad as it sounds, but it is tedious. So the biovolume calculation references, the main ones are in red here. These are the tables that you go to. Let's say you have a Phanezoa Flossaqua or Microcystis aeruginosa. You just go to that table, look up, and see what the biovolume is per cell, and plug it into your cells per mil or natural units. So the first one, algal biovolumes from USGS, from their NAQUA program. And the last one, which is what the Europeans have set up, and this is called biovolumes and size classes of phytoplankton. Either one of those work. I've looked at both of them, plugged in numbers for different genera. In fact, I'll show you an example real quick. And they're very close to the same. So here's an example using our favorite Phanozomenon. So the equation used would be for a volume of a cylinder, right? Because it's a long tapered cell. So we're going to use the cylinder. For Microcystis, it would be a sphere. Here's Anabina or Dolichospermum, same as Phanozomenon, so that's going to be a cylinder. Here's Gliocapsa. You were asking about Gliocapsa. That would be a rectangular box. So the rectangle would work for Gliocapsa. Cylindrospermopsis and some of the Anabinas that are coiled, you're going to use the formula for a screw shape. And that's how you do it. That's how you do biovolumes. A quick reference to epifluorescence. I mentioned that Eric has this Turner little fluorescent probe up here. Chlorophyll A will fluoresce back red under blue excitation, blue light. Green excitation causes phycoerythrin to fluoresce back bright orange, and phycocyanin to fluoresce back orange yellow. So these little fluorescent detectors will pick up those pigments, and they're not expensive. So that's the usefulness of fluorescence. And these are just a couple of microscopy sites. So there's enumeration, biovolume, taxonomy. I'm going to come back to taxonomy just before the lab, because I want to show you specifically the major genera and where they fall in the taxonomic key that are responsible for toxicity. I'm going to do that just before we hit the lab material.